Hello, my name is Sydney Walker, and the title of this poster is Modeling Laminopathies in Drosophila, Comparative Analysis of LMNA Mutations that Cause Muscle and Adipose Disorders. Mutations in the LMNA gene cause a collection of diseases known as laminopathies. Laminopathies include muscular dystrophy, lipodystrophy, and early onset aging syndromes. The primary focus of my research are these two individuals pictured here, Viles and Priscilla Lopes-Schliep. These two individuals live varying lifestyles. While Viles is confined to a wheelchair, Priscilla Lopes-Schliep is a bronze medalist on the hurdles. Viles has a lipodystrophy as well as a muscular dystrophy, while Priscilla Lopes-Schliep only has a lipodystrophy. Emery-Dreifuss muscular dystrophy, or EDMD, causes skeletal muscle atrophy and cardiomyopathy, which Jill has. They both possess Dunnigan-type familial partial lipodystrophy type 2, and it causes a reduced subcutaneous fat in the limbs and an excess of fat in the face, neck, and upper back. We wanted to see what makes these individuals so different from one another. And to understand this, we need to understand the structure of the protein that the LMNA gene encodes. It encodes lamins, which are filamentous proteins that line the inside of the nuclear envelope, and they provide structural support for the nucleus and organized chromatin. We'll be looking at Lamin C, which is a Drosophila ortholog, and it has a head region, a rod region, as well as a tail region. And the mutations of this poster will be located in the tail region, and more specifically in the IG fold. Listed here are the human mutations, which include both Jill and Priscilla's mutation, and the Drosophila ortholog, which I will now be referring to as K521W and R564P, as well as a third mutation that we looked at that is in the same place as Priscilla's mutation, but is a different amino acid substitution, K521Q. We wanted to ask, how do the mutant lamins affect muscle cytology and function, and do the mutant lamins cause heart defects? To do this, transgenic flies were generated possessing the mutant Lamin C, which, again, as I said, is an ortholog to the human LMNA, and using a tissue-specific GAL4 UAS system, the mutants were expressed in the larval body wall muscle as well as the heart. Immunohistochemistry, viability, motility, and cardiac aging assays were performed, and here are the results. As we saw in immunohistochemistry, we saw that K521Q as well as R564P show mutant lamins leaking into or mislocalizing out into the cytoplasm, and K521W has abnormal nuclear shape as compared to the wild type. We also did motility assays on the larvae and saw that none of the mutations cause any notable muscle or motility defects. However, adult viability was significantly decreased, which meant that a lot of these mutations are dying at the pupal stage. We also expressed the mutations in the cardiac muscles and saw that the mutation R564P reduced the adult lifespan significantly compared to the rest of the mutations. So what we can draw from this is that mutant Lamin C mislocalizes and changes myonuclear shape. Larvae expressing mutant Lamin C have unaffected locomotion but very little adult viability, and cardiac-specific expression of the mutant Lamin C decreases adult lifespan in mutations linked to EDMD. From here, we will want to compare lipid droplets in flies expressing wild-type and mutant Lamin C in the muscle and the fat, and measure the changes of gene expression in the muscle. And I would like to thank Lori Walrath, Ben Hines, Margaret Ketterer, and the rest of the Walrath Lab, as well as the NIH and iCrew for funding this opportunity. Thank you.